Hi, my name is Ting Oliver and this is my project, The Effects Nutrients Have on Plant Growth. I originally came across the idea from a packet that my teacher sent me, and it contained ideas of science projects, but I changed the idea to be more specified. The question of this project is, at what level of pH, potash, phosphorus, and nitrogen does a bean plant grow best in? Before I could determine how to proceed with the experiment, I had to research three main nutrients that all plants need in order to survive, in addition to researching pH levels. These nutrients were nitrogen, phosphorus, and potash. I found out that plants need the correct pH level because it controls how well plants utilize the nutrients available in the soil. In addition, I learned that nitrogen helps make amino acids, the building blocks of proteins, and that nitrogen is a major component of nucleic acids. Without nitrogen, plants wouldn't be able to make proteins and the amino acids that make up the plant. Lastly, I learned that growing plants need phosphorus because it helps build plant DNA and helps seed development, and that plants need potash because it is essential for water uptake in addition to making plant sugars for use as food. After doing a bit of research, I could finally proceed with my project and determine the variables. The independent variables I changed were the levels of pH, nitrogen, potassium, and phosphorus in soil by collecting soil samples from different areas. By collecting from different areas, I can ensure that I get different levels of nutrients. A control variable I had was the amount of water each soil gets and the amount of sun. After determining the variables and doing research, I came up with a hypothesis. My hypothesis was, if you plant bean seeds in soil that is too acidic or basic and has deficient levels of nitrogen, phosphorus, and potash, then the growth of the bean plant is stunted and the plant is unhealthy when compared to a bean plant grown in optimal soil conditions. In this project, I used 10 different soil samples from South Carolina, Florida, and different types of bagged soil. I also grew strictly one kind of bean plant, used Jiffy pots to grow my bean plants in, and an artificial light so I could control how much light energy the plants get. Before I could grow my bean plants, I had to test the soil. I bought a soil testing kit from Home Depot that tested all the variables I wanted to test. The soil test tested pH levels, nitrogen, phosphorus, and potash levels. The soil test came with basic instructions that were easy to follow. Basically, you put soil, distilled water, and a powder into a test chamber. You wait 10 minutes and then look at the color of the water. The test chamber has a color chart film that you refer to when determining the pH level. You repeat these steps for every pH test. For testing the nitrogen, phosphorus, and potash levels, you follow similar instructions. The only difference is that you have to put some of the soil into a container or mason jar with distilled water and shake it up, and then let it rest undisturbed for 24 hours. Then, after 24 hours, you follow the same instructions as the pH test. After you shake it up and let it sit for 10 minutes, look at the water and refer to the color chart film on the test chamber to determine the nutrient levels. You repeat these steps for every nutrient test and for every soil sample. After testing the soil, I could finally plant the bean plants. I planted 5 bean seeds in each pot, all relatively in the same location, and I labeled them A, B, C, D on the pot, and E was always in the middle. By doing so, I was able to keep track of all the plants that grew in each pot and measure them every day without getting mixed up. I labeled each pot 1 through 10 with the different soil samples and grouped the plants for 14 days and measured the growth of the plants every day. For 14 days, I used the artificial light to help grow the plants. After measuring for 14 days, I was ready to do some calculations. I determined the average growth rate per pot, the average maximum growth height of plants in each pot, and the germination rate. With these new calculations, I made one big table with all the data. With one big table, I was able to look at my data in whole and determine how to make the graphs. I finally decided to create an X and Y table. The x's would be days to germinate, germination rate, maximum height, average maximum height, and growth rate. The y's would be phosphorus, potash, nitrogen, pH, and total nutrients. So for each graph, I needed to make a graph comparing the y's to each of the x's. After analyzing my data, I came up with a conclusion. In my experiment, I discovered data that does support my hypothesis, but not very clearly due to experimentation flaws and a few errors. My hypothesis was, if you plant bean seeds in soil that is too acidic or basic and has deficient levels of nitrogen, phosphorus, and potash, then the growth of the bean plant is stunted and the plant is unhealthy when compared to a bean plant grown in optimal soil conditions. There were two soil samples that clearly showed how increasing soil nutrients can benefit plant growth. Sample 2 and Sample 10 are the two samples that had the greatest germination rate. Sample 2 from Miracle-Gro potting mix has surplus levels of nitrogen, deficient levels of phosphorus, adequate levels of potash, and is not organic, meaning chemical fertilizers were added. Sample 10 from composted horse manure created on our own farm in South Carolina has surplus levels of all three nutrients, nitrogen, phosphorus, and potash, and is organic. Sample 2 had an 80% germination rate, while Sample 10 had a 100% germination rate. And Sample 2 had an average growth rate of 2.0 centimeters per day, while Sample 10 had an average growth rate of 1.7 centimeters per day. These two data are very similar in terms of growth rate and germination rate, but the difference is that one is organic and one was store-bought. Organic means natural methods and no chemical fertilizers. Sample 2 is not organic because it was treated with man-made fertilizers. The results of these two samples were interesting because they show that organic fertilizers are as good, if not better, than chemical fertilizers. Because people are trying to decrease chemicals in their lives and willing to pay more for organic produce, this shows that farmers could make more money growing organically to increase their crops and sell them for more money. On the other hand, some of my data were unclear because not all the samples with adequate nutrients grew well, leading me to believe that other variables were involved that I did not account for. For example, soil samples 5, 7, and 9 showed no growth, yet their nutrient levels weren't deficient. For example, Sample 5 had surplus amounts of phosphorus, Sample 7 had an adequate amount of potash, and Sample 9 had surplus levels of nitrogen and phosphorus. In addition, the soil samples with no growth also had close to average pH levels. Based on this data, I expected there to be growth, but because there were none, this could mean a human error. I made the mistake of overwatering, and this causes fungi to grow and rot the seeds before they have a chance to grow. In addition, Sample 7 and 9 may not have grown because it was clay soil. Clay soil can be bad for plants because clay compacts easily, making it hard for the roots to penetrate the soil to develop a root system, resulting in stunted root systems. Also, because the clay is very compacted, this allows very little amounts of oxygen to enter the soil, and therefore the seeds become oxygen deprived and won't grow. Overall, the data of my experiment were somewhat inconclusive because some of my data supported my hypothesis while other data did not. My experiment would have benefited from repeated testing of soil samples and reducing error in watering and measurement. I should have also had a way to more clearly quantify soil qualities such as clay, sand, and loam content. In my experiment, there are many errors. The error I made is that I overwatered a few of my plants and caused mold to grow. This is a problem, especially with plants that are just starting out as seedlings, because wet conditions surrounding struggling seeds often encourage harmful fungi to grow. Another error is that I left between days 6 and 9 and had to ask a woman to take over measuring for me. She made the unfortunate mistake of switching between centimeters and inches. Luckily, I was able to look at the data and do conversions for the measurements that didn't seem right, but I could only guess that certain measurements weren't right. This affected the project's end results because not all the data were correct, and I ended up having to guess certain measurements and overall skew the results. I also made the error of using qualitative data instead of quantitative data for my soil testing, and I didn't test the soil enough times. If I sent the soil samples to a professional lab, I would get specific numbers of levels of nutrients instead of words such as surplus or adequate. This would give me a much more accurate result for my experiment and make it more scientific. In addition, I could have tested the soil multiple times to increase the accuracy of my project. To improve my project in the future, I could control the amount of water each pot gets. This way I can make sure I don't overwater, and so each pot has the same amount of water. In addition, I can improve my experiment by collecting more samples from all over the world. By collecting more diverse soil samples, you could get results that are very different from each other. To add on, I can improve my project by repeating the test several times and growing the plants in larger pots. An experiment you could perform as an extension to my project is adding fertilizer to the weakest soil. By amending the weakest soil, you could observe if the fertilizer works to fix and add enough nutrients to allow plants to grow. Another extension I would like to make is to have more time to grow out the beans fully. Growing just the plant doesn't tell you very much about the amount of food it will produce, but allowing it to produce beans will tell you the quality of the soil. This project is meant to inform farmers that produce food, and just showing the initial growth won't tell you the amount of food it will produce, and therefore won't be very informative for farmers. In addition, an extension of this project could be to test the soil again after the plants grow. If we test it again after the plants grow, we could see whether the nutrients in the soil are now depleted because the plants used it or if it remained the same.